Considering the current situation of COVID-19, there is in particular a public interest on increasing immune defense easily. Feeding your body certain foods may help keep your immune system strong. So if you're looking for ways to boost your immunity during this pandemic, then plan your meals to include these 15 powerful immune system boosters. So let's see which are those. Citrus fruits. Most people turn to vitamin C after they have caught a cold. That's because it helps build up your immune system. Vitamin C is thought to increase the production of white blood cells. These are key to fighting infections. And most popular citrus fruits like grape, oranges, lemon. As your body doesn't produce or store it, you need daily vitamin C for your health. Almost all citrus fruits are high in vitamin C. With such a variety to choose from, it's easy to add a squeeze of this vitamin to any meal.
Jai Hind, dear children. Welcome to biology class. Today we are going to continue with the chapter Heredity. Last class we have discussed about some terms such as alleles, genes, genotype, phenotype, dominant gene, recessive gene, monohybrid cross, dihybrid cross, etc., which are used in genetics. And the monohybrid cross done by Mendel. So last class we learned the monohybrid cross by taking height as the character, in which tall and dwarf are its two contrasting traits, and got 1:2:1 as the genotypic ratio and 3:1 as the phenotypic ratio in the F2 generations. And we also discussed that how these traits are expressed in the organisms or the plants produced in the F1 and F2 generations. So today we can learn about the dihybrid cross done by Mendel and the three laws of inheritance postulated by Mendel and how the sex is determined in different organisms, especially in humans. So last class we have learned how to do a monohybrid cross and how to draw the flowchart of a monohybrid cross. Then also I will explain the monohybrid cross once more using or considering the height as the character. Mendel selected pure tall plants and pure dwarf plants for doing a monohybrid cross. The genotype of the pure tall plants is capital T, and the pure dwarf plant is small t, small t. The next step is to cross-pollinate these pure tall plants and pure dwarf plants. And these two are called as the parental generations. At the time of reproduction, meiotic division takes place and the gametes are produced. So there is possibility of two gamete formation in the tall plants. That is, one gamete contain the gene capital T and the other gamete also contain the gene capital T, as this is a homozygous chromosome. And the dwarf plant also produces two gametes, each contains the small t. The next step is the fertilization or the fusion of gametes. So the possible chances of fusion of gametes are one capital T and one small t, that forms capital T small t. Other possibility is another capital T and capital T from the tall plant and small t from the dwarf plant. Another possible fertilization is the gametes containing one capital T from the tall plant and the small t from the dwarf plant. And another possibility is another capital T from the tall plant and the small t from the dwarf plant. So there forms four plants which contains the genotype capital T and small t. Even though these plants contain the recessive gene small t, then also the physical appearance of all these plants F1 generation are tall plants. And even though the small t which shows the dwarfness is there, then also it is masked by the capital T or the dominant gene in the F1 generation. Why? That is why the recessive character is not expressed in F1 generation. Only tall plants are appeared in the F1 generation. The next step is production of F2 generation by self-pollinating the F1 generations. So for that, a male reproductive part and female reproductive part is taken. So the male part produces two gametes, and as it is the heterozygous chromosome or as it is a hybrid plant, it shows heterozygous chromosome. And the one gamete will be the capital T and the other gamete will be the small t. It will contain the small t. These gametes are allowed to fuse or fertilization. F2 generations are produced. Now let us see the combinations or fusion of these two gametes while self-pollinating the hybrid plants of F1 generation. We can do this cross using a checkerboard. On the upper side we can write the male gametes and the left side we can write the female gametes. So the possible fusion are one male gamete containing capital T and another male gamete containing and the female gamete containing capital T forms the capital T. So like that, two capital T small t plants are produced, and as well a plant containing two small t are produced, and that is a dwarf plant. So four plants are produced. Of these, three are phenotypically tall and one is dwarf. So we can write the phenotypic ratio as 3 tall is to 1 dwarf. But what about the genotypic ratio? The genotypes of these four plants are of three types. One is capital T. The other two plants are capital T, small t, and the one plant has the genes small t, which is the recessive character. So we can write the genotypic ratio as 1:2:1. So like this, we can do the monohybrid cross by using another character, that is flower color. Usually pea plants produce two colored flowers, that is purple and white. The purple is considered as the dominant character and the white is considered as the recessive character. So here one pure plant with purple colored flower and pure plant with white colored flowers are selected and allow them to cross pollinate. When these two plants are crossed, the F1 generation obtained are all purple plants. That is, all the plants
So why did the recessive genes
character.
not express themselves
Do you know
in the
why
F1
all these
generation?
plants shows dominancy?
And how did they
in
get
this F1
expressed
generation
in the
of the
F2
monohybrid
generation?
cross? And if these F1 generations are allowed to self-pollinate, they produce the plants with tall plants and the dwarf plants. So why this F2 generation shows this dwarfness or the recessive character? So let us observe
Let's
a monohybrid
understand
cross
this letter P
using the
written
flower
in
color
uppercase
as
as the allele, that is
contrasting character.
factor in Mendel's terms for the purple colored flower. And the white colored flower will be represented with the allele P written in lowercase. Now, the first cross included purple flowered plant and a white flowered plant. So this is how we can represent this crossing. Any idea why have we used the letter P twice here? It's simple. Green peas, there are two sets of genes in it. That is why we use the letter twice to represent both the allelic forms. So here, PP written in uppercase will represent the purple color of flowers, while PP written in lowercase will represent the white color of flowers. Now in this cross, the gametes, which are nothing but the reproductive cells, will be formed like this, and the cross will yield us this result. How? This combination, this one, then this one, and this is the last one. These are the four possible combinations. Now, as you can see here, PP indicates that the offspring has one dominant and one recessive allele. In such a case, the plants will have only purple flowers, as the presence of one dominant gene is enough to express the trait. That is why all the plants have purple colored flowers in the F1 generation. Now, can you guess why the ratio 3:1 is obtained in the F2 generation? Let's find that out with the help of a similar theoretical cross. Here, we take any two random plants from the F1 generation. Crossing these two, or even by carrying out the selfing process in a similar fashion, gives us this result. Now, as we can see here, these three offsprings have at least one dominant gene. That is the reason why each of these is a purple flowered plant. Now in this case, both the genes are recessive, that means no gene to dominate the trait. As a result, the plant expresses the recessive trait, that is, white colored flowers. This was the simplest way to explain Mendel's results. Dominant genes are those which are expressed in both homozygous and heterozygous forms. On the other hand, recessive genes are those which can be expressed only when present in homozygous form. Consider the character stem height in the pea plant. Here we take a tall and a dwarf plant. Both these are pure lines. On crossing the parental generation, we get these offsprings in the F1 generation. Now, what will happen when we cross these two plants from the F1 generation? It will give us these results. Take a moment to go through this. So what we get is 3 tall plants and 1 dwarf plant. Isn't this the same ratio that Mendel obtained when flower color was considered as the character? Yes, it is. He had obtained 3 purple and one white flower in the F2 generation. Similarly, here we get 3 tall and 1 dwarf plant.
And from this observation, Mendel postulated the law of dominance. Law of dominance states that some alleles are dominant while others are recessive. And the character which gets expressed in the presence of its contrasting trait is termed as the dominant trait, and the trait which remains unexpressed in the presence of its contrasting trait is termed as the recessive trait.
It's at least one dominant allele, which means if one or even both the alleles are dominant, then we get the respective dominant phenotype. Now let's cross this F1 generation to check whether we have understood the law correctly. The cross of these F1 hybrids gives us these offsprings in the F2 generation. Can you help me with the phenotype and the genotype of these offsprings? Here we get 3 tall plants and 1 dwarf plant. Genotypically, there is one plant that is homozygous for the dominant trait. There are 2 plants which are heterozygous, and this last one is again homozygous but for the recessive trait. So can we relate these results with our law of dominance? There are 2 types of alleles, one dominant and one recessive. This is what the first part says. And the second part says that at least one dominant allele displays the effect irrespective of the presence of the recessive one. So as we can see here that these 3 plants are tall because they contain the allele T for tall trait. This one has both the dominant alleles, and these 2 have one allele for the dominant trait. Its presence masks or suppresses the effect of the recessive allele.
So this is how Mendel postulated or concluded the law of dominance.
So
Next law is law of segregation. Now let us see how Mendel postulated the law of segregation. The law of segregation states that during gamete formation, the alleles for each gene segregate from each other such that each gamete formed carries only one allele of one gene. This we can understand from a monohybrid cross.
Generation, where the male plant was tall and female plant was dwarf. Since the plants in P generation were true breeding, the 2 allele in each of these plants will be identical according to the trait. So, the male will have 2 dominant allele for tall stem length, and the female will have 2 recessive allele for dwarf stem length. Each of these plants will produce gametes. According to Mendel, alleles separate during gamete formation. So here, we will have 2 gametes formed by the tall parent each having a single tall allele. And dwarf parent will produce gametes each having a dwarf allele. Note that both male and female plant of P generation produced a single type of gamete. When fertilization takes place, the 2 gametes fuse. So, in the F1 progeny we have now 2 different allele. Recall that, when dominant and recessive allele are present together, the recessive allele is not expressed. Since tall allele is dominant, the individuals of F1 generation are all tall. This explains that, in the monohybrid cross, all the F1 progeny had tall stem length. Let's now understand what happens when, in an individual separate independently of each other.
That when gametes
That
are
means
formed,
a gamete
the alleles
carries
of
only one
each
allele
gene
for
in an
each
individual
inherited character.
separate independently
And
of
when
each
gametes
other.
unite during fertilization, each gamete
And
contributes
when these
one
gametes
allele.
unites during fertilization,
Let's understand this by
each
illustration.
gamete contributes one allele.
Recall that,
And
in the
forms the hybrid
monohybrid
plants.
cross we have the P generation.
And on the basis of this observation, Mendel postulated that at the time of reproduction, when the gametes are formed, the genes separate or segregate so that each gamete receives only one allele of each character. And this is called as the law of segregation. Next, we can learn about the dihybrid cross. That means the crossbreeding of 2 individuals of same species. here the pea plants considering two contrasting characters. For example, one character we can take it as the height of the plant or the shape of the seed, or we can consider the shape of the seed or the color of the seed. Like that, two contrasting characters can be considered for doing the cross. Here we can do a dihybrid cross and can learn how to draw the flowchart of a dihybrid cross. Here the two contrasting characters taken are seed shape and seed color. Usually the pea plants produce yellow and green colored seeds and round and wrinkled shaped seeds. So here first the parental generation are taken and appear the plants that produce a round and yellow colored seeds, and another plant with wrinkled and green colored seeds, which is recessive character. Now at the time of reproduction the gametes are produced, and here the dominant plant which contains the round and yellow colored seeds. We can write the genotype as capital R, capital Y, and the recessive plant is small r, small y, that is wrinkled green seeds. Now during reproduction the gametes are produced. So the gametes produced, the possible gametes produced may be capital R, capital Y and capital R, capital Y. And the wrinkled seeds, green seeds producing plants produce the gametes with genes small r, small y and small r, small y. Next step is producing F1 generation, and we can do this in the checkerboard. So on one side the gametes, the male gametes, and the one side female gametes. So what is the possible fusion? Capital R, capital Y fuses with the small r, small y, forming capital R, small r, capital Y, small y. So another two combinations, that is capital R, small r, capital Y, small y, capital R, small r, capital Y, small y, and the other two combinations also capital R, small r and capital Y, small y. So these are the plants produced in the F1 generation. And all these plants, the phenotype or physical appearance of all these plants are round. These plants produce a round and yellow colored seeds. The next step is self-pollinating F1 generations. Now the possible gametes produced from the male reproductive part and the female reproductive part of a flower. There are four possible chances are there, that is capital R, capital Y, capital R, small y, capital small r, capital Y, and small r, small y are four possible gametes. So let us do this cross of these gametes by the help of a checkerboard. So on one side we can write the female gametes, and on one side we can write the male gametes, and we can do the crosses. So the first column or the first gametes, capital R, capital Y, and capital R, capital Y fuses together, then there forms the plant with the genes capital R, capital Y, capital Y. And so we can do all these crosses. So the first capital R, capital Y can be fused with all the other male gamete, and the second capital R, small y can be fused with all the other gametes. Next, small r, capital Y can be fused with all the other four gametes, and next small r, small y can be fused with all the other gametes. So like this we can do the cross. And here how many different types of plants are produced? So the genes, the plants those having the capital R and capital Y. They shows the round and yellow colored seeds. Now those having the capital R and small y, they shows the round and green colored seeds. And those with the small r and capital Y shows the wrinkled and yellow colored seeds. And the small r and small y, genes with small r and small y shows the wrinkled and green colored plant seeds, which are the recessive character. So 4 types of plants are produced. One is round and yellow, 9 plants are produced. Other is round and green, that is 3 plants are produced. And the wrinkled and yellow, that is 3 plants are produced. And the wrinkled and green, 1 plant is produced. So we can write the phenotypic ratio as 9:3:3:1. But the genotypic ratio is different because there are new combinations of genes. So we can write the genotypic 1:2:1:2:4:2:1:2:1. Okay. Understand more about the dihybrid cross from the following video and can understand the third law of inheritance, that is the law of independent assortment.
The 2 characters were seed shape and seed color. Today, we will understand in detail Mendel's law of independent assortment. Let's begin. Mendel crossed a true breeding plant producing round yellow seeds and true breeding plant producing wrinkled green seeds. This was the P generation. Now, we know that, during gamete formation, each gamete will have 1 allele for seed shape and 1 allele for seed color. That's, each gamete will have 1 allele for each character. So, the gametes produced by the plant with round yellow seed will contain dominant R and Y allele. Similarly, the gametes produced by the plant with wrinkled green seed will contain recessive R and Y allele. In the F1 generation, Mendel obtained all plants with round yellow seeds. If we look at the genotype of the F1 plant, we know that dominant R and Y allele came from one true breeding parent, and recessive R and Y allele came from the other true breeding parent. To highlight this fact, I have used 2 different colors to represent the genotype. Now, Mendel's question was whether the allele of different genes are transmitted together during gamete formation, in the same combination that original parent had. In other words, will dominant R and Y allele always be transmitted together generation after generation? And, will recessive R and Y allele will be transmitted together? Or, the allele of different genes are transmitted, independently of each other. Heredity states that allele of different genes do not stay together when gametes are formed. In other words, allele of different genes behave independently of each other. Let's see how gametes will be formed according to this possibility. Each allele for a character can pair with any of the allele for the second character. For example, allele responsible for round seed shape can pair with allele for yellow seed color, as well as green seed color. Similarly, allele responsible for wrinkled seed shape can pair with allele for yellow seed color, as well as green seed color. So, there are 4 possible combinations of allele for these 2 characters. Or in other words, 4 types of gametes. Since both the male and female plant of F1 generation have the same genotype, both will produce the same type of gametes. Let's now draw the Punnett square with 4 rows and 4 columns. Write the gametes produced by male parent along the top of the square, and write the gametes produced by female parent along the left side of the square. Now, write down the combinations of allele in the offspring, that is, F2 generation, in the boxes of this square. From Punnett square, we find that there will be 9 round yellow seed producing plants, 3 round green seed producing plants, 3 wrinkled yellow seeds producing plant, and 1 wrinkled green seeds producing plant. The ratio is 9:3:3:1. Here, you must have noted that 2 new phenotypes were also obtained. Plants with round green seeds and plants with wrinkled green seeds. Mendel obtained same results in his The Hybrid Cross experiment. The trait.
Therefore, he gave his third law. Law of independent assortment states that when 2 or more characteristics are inherited, individual heredity factors assort independently during gamete production.
The next topic we want to study is the sex determination. Sex determination is the process by which the sex of an individual is determined. Sex determination can be done by 2 ways. One is through genetics or genotypic method, and the another is by environmental factors. That is, one is genetically and the other one is non-genetically. First, we can discuss about the non-genetical method of sex determination. Non-genetical sex determination, that means with the help of environmental factors. So, in some animals like turtles, alligators and crocodiles, the temperature at which the fertilized eggs are kept determines the sex. If the temperature increases, some animals hatch out as male, and if the temperature decreases, some animals hatch out as female. And some organisms such as snails can change their sex even. Sex of some animals depends on the environmental factors such as temperature. That is, the sex of the offspring depends on the temperature at which the eggs are incubated. That means the temperature for which the eggs are to be hatched. So, the sex determination of such animals are temperature dependent, such as crocodiles, alligators, etc. If the temperature is high, the female offsprings are produced, and if the temperature is low, the male offsprings are produced in such animals. And in case of turtles, if the eggs are incubated above 32 degrees Celsius, the females are produced. And the eggs are incubated below 28 degrees Celsius, the males are produced.
Sex of a child is determined by the sex chromosomes.
The genotypic method of sex
We know
determination.
what sex chromosomes are.
The determination of sex based on the sex chromosomes.
You may see some thread-like structures in it. These are chromosomes.
In humans, for example, the last pair is the sex chromosomes, while the first 22 pairs are called autosomes. Similarly, in many other organisms, the sex chromosomes help to decide the sex of an individual.
There are 23 pairs or 46 chromosomes in a human cell, out of which 22 pairs, that is 44 chromosomes, are autosomes, and 1 pair is called sex chromosomes. These sex chromosomes carry instructions for sex determination. Sex chromosomes are of 2 types, X chromosome and Y chromosome. In a female, only 1 type of sex chromosomes are there, which is X chromosome. But in a male, both X and Y sex chromosomes are there. Let us see the arrangement of chromosomes in an unfertilized egg. An unfertilized egg has 22 autosomes and 1 X chromosome, whereas sperms have two types of arrangements. One type has 22 autosomes and one X chromosome. Another type has 22 autosomes and one Y chromosome. Now we see how the sex of a baby is determined. When an egg is fertilized by a sperm containing X chromosome, the zygote formed would have two X chromosomes. This zygote is developed into a female child. When an egg is fertilized by a sperm containing Y chromosome, the zygote formed would develop into a male child.
If we looked at this in a Punnett square, the gametes from the female would always be X, so XX, and the gametes from the male would be either X or Y. Can you complete the Punnett square to show the probability of having a male or a female child? Pause the video, complete the Punnett square and click play when you're ready to check. Did you get it right? So the X from the ovum and the X from the sperm would produce a female baby, and the X from the ovum and the Y from the sperm would give us a male child. So really, it is the man who determines the gender of the child. He brings the Y, which is the only chance of a male child.
3. Sex of the offspring is determined by chromosomes called sex chromosomes. In humans, X and Y chromosomes determine the sex of the offspring. Females have X, and males have X and Y chromosomes.
So today we studied monohybrid cross, dihybrid cross, 3 laws of inheritance and the sex determination in humans and some other organisms. So children, I hope you all understood the contents that we have discussed today. What we have discussed today? The dihybrid cross done by Mendel and the 3 laws of inheritance postulated by Mendel, that is the law of dominance, law of segregation and law of independent assortment, and how the sex is determined in humans and other organisms. So now read the textbook thoroughly and prepare the notes. So by this class, biology portions are over. Now from the next class onwards, we can start the revision. Thank you.